Hi, everybody. This is Dr. Kurt Woeller for Integrative Medicine Academy. This is the Great Plains Laboratory monthly sponsored webinar, complimentary webinar sponsored by Great Plains. I'm really excited about this particular topic for this month. As you know, I've been involved in clinical education for many years. I do a lot of these webinars through Great Plains, and I've been teaching the organic acids test seminar for many years as well. So this particular talk is going to take the organic acid test to a different level, okay? And this is the reason I titled it Advanced Topics in Organic Acid Test Assessment, is to kind of go beyond the basics and be able to highlight a few, I think important things to consider from a clinical standpoint. For those of you who don't know me, I've been an integrative and functional medicine physician for over 20 years. I've also worked with the autism population, implementing biomedical therapies for autism for about that same amount of time. I do a lot of clinical education for Great Plains Laboratory. I also am the speaker both throughout the US and internationally. I've written some books, clinical educator, as well as a practicing clinician. And I started an online training website called Integrative Medicine Academy about 3 years ago. I'm a co-founder of that. I'll talk a little bit more about Integrative Medicine Academy a little bit later. And then, as I mentioned before, I work with autistic kids. I work with people with autoimmune, gastrointestinal, and neurological disorders. So the reason you're here, right, is to hear more about the organic acid test from Great Plains Lab. I think one of the best ways to view this particular test is it's more than just a single test. It's really a series of multiple tests. Each section within the organic acid test could be broken out into its own test. So it's really a very comprehensive profile that helps us to identify markers that can indicate various clinical imbalances for patients with a wide variety of health disorders. As you know, I've been involved in teaching the organic acids test seminar for the GPL Academy. These are weekend seminars on the implementation of the organic acid test into practice. And then there's additional lectures as well on toxicity and chemicals and mold toxicity. If you've not been to a GPL Academy event, I'd highly recommend you go to gplworkshops.com. and you can find out if there's going to be a seminar coming your way in a particular city. So we typically do an organic acid test workshop about every 6 weeks, and so they're quite popular and I think overall highly effective. And the goal of the OAT workshops that are done through the GPL Academy are really an introduction to the organic acid test, in how to think about it, how to implement it into practice, and get started by understanding, the different sections on the OAT. Integrative Medicine Academy is my own academy. We have a wide variety of courses online. The first course I ever started with was my Autism Mastery course. We've expanded it to hormones, we've expanded it to toxicity, and even an entire course on small intestinal bacterial overgrowth. We're just in the midst of launching our second edition of a functional medicine mastery course. I believe this is the most comprehensive functional medicine mastery course out there. It's really meant to take any practitioner who's looking to implement functional medicine into their practice from a foundational standpoint, but we also get into advanced concepts within this course as well. This course just started. There's still spots available. If you're interested, you can go to functionalmedicinemasterycourse.com. And then I'm launching a new course in October called the Advanced OAT Mastery course. So this particular course is all about the organic acid test on an advanced level. If you are a new practitioner to using the organic acid test, or you've not been to a GPL Academy event or heard the basic OAT seminar, and you're brand new to this material, this course is probably not right for you at the time. This is advanced material. If you've been working with the organic acids test in practice and you're comfortable with it, if you've attended some of the GPL seminars, then yeah, the advanced oat is a great segue for you. I actually just noticed here I got a typo. It's Advanced Oat Mastery Course, not .com, not Advanced Oat Mastery Course. My eyes didn't catch that. So Advanced Oat Mastery Course. And then we have a special report. If you text the word OAT, O-A-T, to 66866, you'll get access to this special report. There was a notice that went out earlier today. The link showed up that was working, but apparently the web page that was hosting the book had mysteriously disappeared. It's been fixed, so it's back online. It's kind of the way things go sometimes with internet computers. So you can text the word OAT, O-A-T, to 66866 for this complimentary special report. The running theme of all of my teaching and my courses is to really get people to think critically and think clinically about what you're dealing with regards to these lab tests. The organic acid test, particularly because it's so comprehensive and complicated, you know, we want to move beyond just treating people based on the numbers on the test, but again, to think about things clinically and critically and learn how to prioritize the information based on the clinical presentation. So when we look at a general oat, right, it's got multiple sections. It's got a yeast and fungal markers that indicate invasive candida and mold exposure, bacterial markers that could relate to irritable bowel or even small intestinal bacterial overgrowth, the Clostridia bacterial markers, which have high correlation to digestive system problems, but also brain and neurological issues because those toxins interfere with the nervous system. Oxalate metabolites for kidney stones and chronic pain linked to oxalic acid production. And then there's this whole category of mitochondrial markers to evaluate for mitochondrial dysfunction. And one of the things I talk about in many of my courses, as well as the introductory oat course, is that many things that show up on the oat, from fungal markers to Clostridia bacterial toxins to oxalates, can all negatively affect mitochondrial function. There are sections that look at different metabolites linked to neurotransmitter metabolism: dopamine, norepinephrine, tryptophan, serotonin. We'll talk more about some of these things here coming up. The pyrimidine metabolites is a way to assess for folate metabolism. I'm going to get a little bit more into that here in this particular talk, about how that has a greater impact on methylation. The ketone fatty acid oxidation sections, these fatty acid metabolites have a strong connection to the overall function of the mitochondria. Various nutritional markers, indicators of detoxification, particularly glutathione status, is important because glutathione is a major antioxidant that protects our mitochondria against toxicity. And then on the last page of the oat is these amino acid metabolites, and these are really meant to reveal what are called inborn errors of metabolism, mostly seen in children. But there are some things that show up, which I'll mention here, that could have clinical correlation for some of your patients on a more, advanced topic basis. So let's get into it. So if you've been using the organic acid test for a while, you know that just recently there was a new format that came out for the oat, probably about 6 weeks ago, where a few additional markers were added, and specifically in the neurotransmitter metabolite section. Great Plains added a marker called DOPAC, D-O-P-A-C, and they also removed this quinolinic 5-HAA ratio, primarily because each individual marker of quinolinic and 5-HAA is already there, but they felt as a lab it didn't really have a lot of clinical correlation. So the main thing that's really shifted from a topic standpoint to talk about is DOPAC. So here you see an individual that's got elevated DOPAC, okay, linked to the phenylalanine and tyrosine metabolite section. And specifically, it's linked to the metabolism of dopamine. Let me get a colored pen here. So here is our dopamine, okay? And in order to get to our homovanillic acid, dopamine passes through DOPAC, okay, in order to get our HVA. And we've got these regulators here. We've got catecholamine O-methyltransferase enzyme and then S-adenosylmethionine, which is called SAMe, that help in this process. And so we want to look at, from a laboratory standpoint, but also start to decipher when things are low or high, what may be going on, because there's a lot of moving parts in this particular scenario. Dopamine can be influenced by B6 deficiencies. Dopamine can be influenced because of a SAMe deficiency, or perhaps a polymorphism in our catecholamine O-methyltransferase. SAMe, or S-adenosylmethionine, is part of the methylation cycle. In fact, they actually call this part of the methylation cycle the methionine cycle. And if we take a, sort of step back and take a look at this slide, and we see that homocysteine, if we were to look at this as a clock face, homocysteine sits at our 6 o'clock position, methionine sits at the 12 o'clock position. And so basically we convert homocysteine to methionine through two routes. One is called the short route, where we're converting betaine or trimethylglycine into dimethylglycine. And that's really kind of a direct route up to methionine. And then there's the long route, where we move homocysteine through the methionine synthase enzyme, and we need B12 in order for that to happen. And that particular link also then links us up to the folate cycle. In order to have proper methylation, we have to have good conversion of homocysteine to methionine, and then back downwards from methionine back to homocysteine. And part of the process of making that happen is through the production of S-adenosylmethionine. And SAMe is actually involved in providing substrate for DNA and RNA regulation, for phosphatidylcholine support, for cell membrane health throughout the body, as well as neurotransmitter support. We also, when we're looking at the methylation cycle, can't forget about the transsulfuration cycle, because the transsulfuration cycle helps us to produce glutathione, that very important cellular antioxidant that's going to help with detox, but also help protect our mitochondria. So this is just another view of S-adenosylmethionine involved in the methylation cycle. And again, it's going to support serotonin and norepinephrine and dopamine and DNA function and protein development, phospholipids of the cell membrane. So you can see if we've got a SAMe deficiency, there's a number of things that could be adversely affected. So what is it if DOPAC is high? What are some things to consider clinically? What may be going on here? Well, again, there's a lot of moving parts, and it gets fairly complicated fairly quickly. Dopamine beta hydroxylase is an enzyme that converts dopamine to norepinephrine. If dopamine beta hydroxylase is being inhibited somehow, that could affect dopamine metabolism, and could lead to an increase of DOPAC seen on the test. Now, we can have polymorphisms in the dopamine beta hydroxylase, basically a genetic abnormality. Clostridia toxins can inhibit it. We can also have deficiencies of vitamin C or copper, which might affect it as well. We could have a deficiency in SAMe, so not having enough SAMe could lead to a problem of showing up with elevated DOPAC. And then magnesium deficiency or just increased consumption of tyrosine and phenylalanine could also play into this. If you've heard me talk before on these webinars or seen me in person, you've likely seen this slide before. This is the slide that shows us the relationship between Clostridia bacterial toxins and its impact on dopamine metabolism. So Here we've got Clostridia difficile, and up here we've got multiple species of Clostridia that produce a chemical compound called HPHPA or 4-creosol. Both of those markers are available on the organic acids test. Now, when those markers are high, there is a good chance, let me get a different color pen, that our dopamine beta hydroxylase can become inhibited. If we inhibit dopamine beta hydroxylase, we're not going to get good conversion of dopamine to norepinephrine. That can affect attention, focusing, memory formation, and if we don't get good conversion to epinephrine, it also can affect energy. If we inhibit dopamine beta hydroxylase, you'll start to get a rise of dopamine, which then can also cause an elevation of HPA and also cause our DOPAC to become elevated. So this is a scenario to look at on the test, is what could be inhibiting dopamine beta hydroxylase? Well, we know that Clostridia bacteria can do it. So dopamine beta hydroxylase is an enzyme that is inhibited by these Clostridia toxins. It turns out too, though, that vitamin C and copper are cofactors for dopamine beta hydroxylase activity. And there's been a lot of research into this, that deficiencies in copper and deficiencies in ascorbic acid could decrease dopamine beta hydroxylase activity, therefore increasing the accumulation of dopamine, increasing the accumulation of HVA or DOPAC. And this is common, this is actually more common than people realize, is that a lot of times people are deficient in ascorbic acid, and if they do have a copper deficiency as well, it will impact the function of that enzyme. So one of the things you can think about clinically on the organic acid test is if you've got elevations of the HVA, or if DOPAC is elevated, for example, something to consider is just additional vitamin C, or you might need to do a test to see if somebody's actually copper deficient. Another reason that DOPAC can be high is a polymorphism of the catecholamine O-methyltransferase enzyme, or COMT. Most of the mutations, or I should say polymorphisms of COMT, cause the enzyme to slow down. So when that enzyme is not functioning properly, okay, so in this particular example, if COMT is not functioning well, we can start to get an increase of DOPAC. Now, that would have to be figured out by doing additional testing. Great Plains actually has a DNA methylation panel. Doctors Data carries a similar profile as well. And this is where you start layering in the genetics on top of the information that the organic acid test provides, and which is something that we will get to with certain markers within the advanced oat mastery course. Another marker, another genetic marker, is MAOA, or a mutation in the MAOA that again affects the overall function of MAOA, because that too might impact DOPAC. In that particular scenario, however, the levels would tend to be low, right? Because MAOA is necessary to convert dopamine to DOPAC. If this activity is low, then we're not converting enough, and our DOPAC many times will show up to be low as well. It's a little bit complicated because in this particular scenario, you've got MAOA in a couple different locations. So it's not as easily straightforward to say it's just purely from an MAOA mutation, but that is something to consider. Again, it would need to be tested for individually based off the genetic testing. Well, one thing I want to focus on here is a chemical called Tetrahydrobiopterin, BH4, because BH4 is intimately involved in the methylation cycle, as well as balance and metabolism of neurotransmitters, and the link that it has overall to the entire system. So oftentimes we tend to hyper-focus on a few enzymes, or we'll hyper-focus on, let's say, the methylation cycle, and sometimes forget about the folate cycle or this thing called the biopterin cycle. And as this particular slide highlights, there's a lot of overlap, but this biopterin cycle has a significant influence on neurochemical balance. The MTHFR gene, a lot of people focus on the C677T, and that's good because it certainly has a very strong influence on folate metabolism. What sometimes gets lost in the shuffle is the A1298C version of MTHFR, because this has an influence on tetrahydrobiopterin metabolism. And tetrahydrobiopterin, or what's called BH4, is really important because it's a major influencer on neurochemical production. So again, the DNA methylation panel could be used, or this information, these SNPs, for example, can be tested in other ways from other types of lab tests. This is just an example, but this is a way now to start looking at things biochemically on the oat, and then layering in the genetics to see how the genetics may be influencing some of these enzyme pathways. If we come back to this particular slide, we've already talked about the impact of Clostridia bacteria and how these particular toxins for creosol and HPHPA might influence dopamine beta hydroxylase. But it turns out that BH4, tetrahydrobiopterin, plays a role as a cofactor in conversion of phenylalanine and tyrosine in these metabolic pathways as well. It starts with this particular chemical called a pterin. These things are called heterocyclic compounds where they contain different ring structures. Folate is actually a derivative of a pterin chemical structure. And these pterins basically complex together to call what are called biopterins. And biopterins are linked to tetrahydrobiopterins as these particular chemicals start to accumulate together to form these very complex structures. The bottom line is these biopterin type chemicals act as cofactors in the conversion for the production of dopamine, for serotonin, for norepinephrine, for epinephrine, and even nitric oxide production. Tetrahydrobiopterin, or called BH4, is a cofactor for phenylalanine hydroxylase that converts our L-phenylalanine to L-tyrosine. I'll show you a different slide here coming up. It's a cofactor for tryptophan hydroxylase. That helps us convert L-tryptophan to 5-hydroxytryptophan. Tyrosine hydroxylase for eventual production of L-dopa, and then again, it's also involved in nitric oxide production. Like a lot of these enzyme systems, these are recycling enzymes, meaning it goes from an active state to an inactive state, or an oxidized state to a reduced state, and it's constantly in flux. And anytime you have these recycling enzymes, they're vulnerable to oxidative stress. So that could be endogenous toxins, exogenously produced pro-inflammatory cytokines, for example, could potentially negatively affect these areas. In order for tyrosine to be converted to the catecholamines, we need to have good function of tyrosine hydroxylase plus the addition of BH4. In order to produce serotonin and eventual production in the melatonin, we have to have tryptophan hydroxylase activity plus BH4. And then phenylalanine gets converted to tyrosine because of BH4 as well. Now think about this clinically with patients you may be working with. Phenylalanine gets converted to tyrosine. Tyrosine, one of the things it's important for is supporting thyroid function. Tyrosine also becomes DOPA to support dopamine. Tryptophan becomes 5-hydroxytryptophan to support serotonin and eventual melatonin. And then, of course, we've got things that regulate ammonia and nitric oxide for cardiovascular health, et cetera, that are a part of this too, as well as function of the cell membranes, which these areas support too. So phenylalanine gets converted to tyrosine in the presence of BH4. It's interesting that BH4 is also supported by this chemical up top called nicotinamide adenine dinucleotide. This is another cofactor chemical. It's another recycling or what's called a redox reaction type of chemical. It moves from an oxidized to a reduced state. Very much involved in mitochondrial activity, particularly the Krebs cycle. So it's a precursor to DOPA. If we cannot convert things over to tyrosine, we're going to run into a problem of dopamine deficiency. Tyrosine is a precursor to melanin for pigment formation of our skin. It's a precursor to CoQ10. Where have you heard that before? Well, CoQ10 is active within the electron transport chain of our mitochondria, so it supports mitochondrial activity. I already mentioned that tyrosine supports thyroid function. It also has a regulatory effect on various enzyme systems throughout the body. So if these systems are being blocked, it could be that we've got a deficiency of BH4 because we've got some kind of mitochondrial problem, affecting the recycling of BH2 to BH4. Or perhaps we've got an enzyme block between phenylalanine and tyrosine. What you can start to see on the organic acid test is an accumulation of certain chemicals, particularly phenylpyruvic and phenylactic acid. These are picked up on the organic acid test under the amino acid metabolites on the last page. Now, clinical suspicion of poor BH4 support of tyrosine production could be low thyroid. So anybody who's manifesting with low thyroid, you can look at the organic acid test to see if there's any indication there, areas that you might support. Certainly low energy, well, that could come about because eventually of low epinephrine production, depressed mood, focus problems, or just picking up on the organic acid test like low dopamine, low norepinephrine, and low serotonin. High ammonia, although it's not absolutely definitive of just a BH4 problem, but a lot of times you'll see on the organic acid test a marker called orotic acid or orotic that indicates some ammonia excess, because high ammonia states can be linked to imbalances in this system as well. It's not uncommon to see people who have low mood disorders, focusing problems, attention issues, low energy, have the low serotonin metabolites as well as the metabolites of norepinephrine and dopamine also being low. So this particular example of a test, we have to, again, think critically and clinically about what may be causing these problems and learning how to use the ode in a different way by digging deeper into the interpretation and starting to understand the biochemistry at a deeper level as well. So a defect in the phenylalanine hydroxylase enzyme will certainly cause an accumulation of phenylalanine that you can start to see as a spillover effect of increased phenylpyruvic acid and phenolactic acid on the organic acid test. BH4 is a cofactor to make that reaction happen. BH4 you can actually get now as a supplement. You can also get it as a medication for phenylketonuria when the situation is really, really severe. So on the organic acid test, on the amino acid metabolite section, phenolactic, phenylpyruvic give an indication of some kind of blockage in that phenylalanine hydroxylase enzyme, so you're not getting good phenylalanine to tyrosine conversion. Now, in these particular examples, the levels are just slightly elevated, so it's quite possible that particular enzyme system is not being supported by enough BH4, or it may be that there's some other chemical that might be interfering, with the process as well. This certainly wouldn't be at the level that would be highly concerning of somebody who might have, a serious metabolic disease. But when we're working with a lot of kids or teenagers and adults, we're not always in a disease state. We're looking at functional imbalances of areas that we can make improvements to, and to see how that might correlate to just overall clinical improvement for somebody's condition. And when you're talking about phenylalanine to tyrosine imbalances, that could affect the nervous system. In extreme circumstances, with phenylketonuria, where the levels of phenylalanine are extremely high, it can lead to seizures and mental retardation and other metabolic imbalances. But again, that doesn't necessarily have to, it's not an all or none phenomenon. You can still have imbalances that might affect people from a neurological standpoint. It's interesting in that we know that tyrosine supports melanin. They had a misprint on the slide, they said melatonin. So light skin, people who are pale, who don't tan easily, that certainly could be one thing to consider. But I want to expand upon this a little bit to kind of bring it back to things that I've talked about before in other lectures, is that tyrosine, yeah, it's going to play a role in melanin. It's going to play a role in catecholamine production. It also plays a role in the production of fumarate acetoacetate. Which brings us back to the level of mitochondrial activity in the Krebs cycle. Acetoacetate supports acetyl coenzyme A production. Acetyl coenzyme A production is the entry point into the Krebs cycle, or the main entryway, I should say. Fumarate also helps to enter the Krebs cycle as well at the level of fumarate. So we know that the Krebs cycle is necessary to create the byproducts, particularly our nicotinamide adenine dinucleotide that enters the electron transport chain for ultimate production of ATP. So when you expand things out for what the organic acid test provides, you can start to understand the biochemistry and get an image of this. You can start to see the layering effect that this particular test has and how that might play out for patients that are dealing with fatigue issues, mitochondrial dysfunction, neurological problems, etc. So phenolactic and phenylpyruvic are two markers that are seen in the amino acid metabolite section of the oat. Another marker that you see from time to time is mandelic. Now again, it could be linked to taking phenylalanine as a supplement, or if it's really high, it might be linked to the phenylketonuria, to the PKU, which is going to be more of an issue in a child. But there's another reason that mandelic can be high, is that it's been shown to be linked to styrene exposure. So when you learn how to use the organic acid test, you understand that it provides core material that is clinically relevant right from the get-go in your practice. But it also becomes a test that can help identify certain things that would allow us to start reaching over to other tests to try to fill in the gaps. And what I mean by that is that the organic acid test provides certain markers that are highly suggestive of other problems like chemical exposure. So an elevation of mandelic may be occurring because somebody has a styrene exposure. Styrene is found in plastics and building materials and car exhaust fumes. So the GPL tox test becomes a complementary test to the organic acids test. 4-hydroxyphenylactic, again, found on the last page of the OAT under the amino acid metabolites section. This is actually linked to what's called tyrosinemia. And tyrosinemia from a metabolic disease standpoint are quite serious. Again, something that's usually manifesting in children. There's 3 different types. There's tyrosinemia 1, 2, and 3. Tyrosinemia 1 is the most severe. These kids have just a failure to thrive. Type 2 is we get mental development problems, skin problems. And then the rarest form is tyrosinemia 3, where there's intellectual disability and seizures. But I want to bring something up here because one of the things that, again, tyrosine leads to is support of mitochondrial function, okay, through acetoacetic acid and fumaric acid. So it has a direct route to supporting mitochondrial activity. But if our tyrosine is being blocked somehow and we start to have an accumulation of tyrosine, well, that, you know, can show up sometimes on the organic acids test. We know that tyrosine gets converted to L-DOPA. L-DOPA gets converted to dopamine. Then we have this homovanillic acid as a byproduct of dopamine metabolism. And in this particular scenario, we would also expect, okay, to see likely an elevation of DOPAC. So that would be a normal way that this would go. What happens if you took the scenario of tyrosine, okay, and let's say that this particular pathway is being blocked, okay, maybe it's a BH4 deficiency, and all of a sudden we've got increasing levels of tyrosine. Or what happens if you start taking tyrosine supplementation in large amounts, and we're sitting with somebody who is in the presence of elevated Clostridia, particularly Clostridia difficile. Well, the Clostridia is going to grab onto that tyrosine. Not only are the colonies of Clostridia going to go up, but 4-creosol is going to go up, and the inhibition of dopamine beta hydroxylase is going to go up, which is going to cause our dopamine levels to rise, which is going to cause our HVA levels to rise as well. Now, I'm not going to get into the neurotoxic effects of dopamine, but I'll just kind of say at this point, dopamine is a very reactive chemical within the nerve cell. One of the things it'll do is it'll break down into a chemical that will essentially rob the nerve cell's level of glutathione. And whenever you rob a cell of glutathione, one of the areas, there's many, but one of the areas that is highly compromised or highly vulnerable is our mitochondria. You've probably seen this kind of scenario where you look at an organic acid test and our Clostridia markers are normal, but somebody has elevated homovanillic acid. You have to ask the question, what's going on? What are some possibilities? Well, you could have a dopamine beta hydroxylase mutation. Okay, Great Plains actually has a test for that now to look for a genetic defect in dopamine beta hydroxylase. You might be dealing with somebody who has a vitamin C or a copper deficiency. It's quite possible that they may just be consuming high amounts of L-phenylalanine or tyrosine as well. So you have many times have to sometimes go back and ask the patient what they're taking, or look at many other sections on the organic acid test to try to figure out why. And that's one of the things that we do in the introductory OAT seminar through the GPL Academy, is look at some of these basic factors and try and prioritize the information. Something else I want to talk about is the folate and pyrimidine methylation link. The organic acid test has a couple markers that are indicative of folate metabolism called pyrimidines. Oftentimes you'll see either uracil and or thiamine elevated in people who have folate deficiencies. It is elevated in certain cancers and genetic disorders as well. And it's interesting because these pyrimidines, mainly thiamine and uracil, are strongly reflective of DNA and RNA activity. Thiamine is specific to DNA, uracil is specific to RNA. What you will most commonly see in an organic acid test when one of those pyrimidine markers is elevated is that uracil tends to be more elevated than thiamine. And I think the reason that that's happening is that uracil is linked to RNA. And so there's just a lot more activity at the RNA level than there is at the DNA level through RNA synthesis, and then the translation effect to protein development. So there's a lot of metabolic demand for adequate folate metabolism at that area. We could take it deeper. We know that the folate cycle is linked to the methylation cycle, and that there are people who have genetic abnormalities that affect the way that their body converts inactive folate to folate. We know that folate metabolism also influences function of the DNA and RNA. It has an influence on ATP, so it has an influence on mitochondrial activity, and interestingly enough, it has an influence on nicotinamide adenine dinucleotide levels. If we were to take this all the way back, we know that NADH not only supports mitochondrial activity, but NADH also is a recycling chemical for BH2 to BH4 conversion. And we need the BH4 to help in the scenario we just talked about of phenylalanine to tyrosine conversion. This particular slide also comes from a lecture that I've done on autism, and particularly looking at attention issues. Folate is also linked to the dopamine receptor cycle. It's not really a cycle, but it's linked to dopamine receptor activity, which is then linked to attention deficit. It's been linked to schizophrenia, bipolar, and eating disorders as well. So it's not just the methylation cycle, it's not just the folate, it's not just the biopterin. All of these things are interacting. There's an overlap between all of this. But if we think about what's happening clinically with many people with chronic health problems, is when we do start to have an issue in the methylation cycle, one of the things that potentially becomes compromised is glutathione. I don't know why it's not switching here. There it is, glutathione. So go to the indicators of detoxification section on the organic acid test. Pyroglutamic is a marker of glutathione status. The reason that these two markers have an asterisk marker next to them is that they're what they're called is indirect, meaning that when the marker is elevated, it indicates a deficiency of glutathione. Pretty straightforward with pyroglutamic. High pyroglutamic indicates a glutathione deficiency. The 2-hydroxybutyric is not so simple. Okay, there's a number of factors that can lead to its elevation. So, but the question you have to ask yourself is, if you see an elevation of 2-hydroxybutyric, is why? Why is it high? Is it because there's a glutathione deficiency? Is there some kind of methylation problem? Is there a transsulfuration issue? Because there's a lot of moving parts with regards to 2-hydroxybutyric. The most common reason is that the body is under demand for increased glutathione. The body wants more glutathione as an antioxidant. But then you got to ask the question, well, why is that happening? Does the person have fungal problems? Do they have Clostridia? Do they have high oxalates? Is there molds? Is there chemicals? So you're always asking, what's the next question? Where else do I perhaps need to look? Perhaps we've got an issue within the methylation cycle. Maybe there's a deficiency of methylcobalamin or folate. There could always be genetic variants occurring in different steps within the methylation and transsulfuration pathway as well. Somebody who's diabetic may show elevations of 2-hydroxybutyric. Somebody who drinks excessively may as well. So again, questions you may need to ask, and looking at your clinical history and the clinical intake for a patient based on this particular marker. So the methylation and transsulfuration pathway are linked. If we were to draw a line right underneath homocysteine, above the line is our methylation cycle. We have the folate cycle over here, and the link between the two is right here, methionine synthase. Methionine synthase is what's responsible for converting homocysteine to methionine through the long route of methylation. But we have to have good B12 status, particularly methyl B12 status, in order for that to occur. Below the line is our transsulfuration pathway. And we know as we move down from one step to the next, we ultimately come at the level of cysteine. Now, cysteine can support sulfation for liver detoxification. It can support taurine and eventual bile salt formation. Bile salts are important for digestive health. We need good bile salt production from the liver to activate the pancreas to release digestive enzymes. We need bile salts in order to activate motility of the small intestine through what's called the migrating motor complex. We have to have bile salt activity even within the large intestine. One of the other components that gets produced is glutathione. There are many things that can lead to glutathione deficiency: toxic chemicals, Clostridia, nutritional deficiencies, oxalates, mycotoxins. In fact, I could take this same slide and scratch out glutathione deficiency and write in mitochondria. Mitochondrial dysfunction could be caused by all of these things too. One of the most common reasons that we see 2-hydroxybutyric elevated is because the body is under demand to maintain glutathione levels, so we get this excessive shunting effect. As things are moving down, we're shunting over towards glutathione with an increase of 2-hydroxybutyric. There's some additional points of interest I want to go through here as we kind of summarize and wrap this information up. Most of the time when you look at an organic acid test, you look at the N-acetylcysteine marker found under the nutritional markers section to be either normal or it's kind of on the low end. Sometimes you'll get a marker back that's high. Now, it could be, and you have to ask your patient, you have to look at their supplement list, their level of N-acetylcysteine might be elevated because they're actually taking a supplement of N-acetylcysteine. That's always a possibility. More times than not, the N-acetylcysteine marker is normal. Another reason that the N-acetylcysteine might be high is that there's a conversion problem that's occurring. There could be just a conversion problem where cysteine is not being converted into glutathione. So it could be because of an enzyme block. What's interesting too is that mitochondrial dysfunction, or excuse me, mitochondria make ATP. And we need adequate ATP in order to make glutathione. But if we have a mitochondrial dysfunction that perhaps is affecting the way we produce ATP, that ultimately could affect the way that we produce glutathione, which then further worsens mitochondrial dysfunction. I know some of you who've done OAT testing have seen this scenario, the fatty acid metabolite section. Remember, the fatty acid metabolites have a strong link to mitochondrial activity because fat is one of the main ways that we burn energy to produce a high amount of ATP. Oftentimes, this particular area of the test is influenced by bacteria, by yeast overgrowth. It can also be influenced by diet. Many times people who are on a ketogenic diet or just a high-fat diet, you'll oftentimes see all of these markers elevated. But when you start running into scenarios where you get the first two markers high all on their own, you kind of have to ask the question, why are these two elevated? Okay, a common scenario is malabsorption. Could also occur because of somebody who's either diabetic or pre-diabetic. You may be just dealing with somebody with really bad digestion, and so they're, again, they're not absorbing things effectively. And this is exactly where this particular scenario came from. We don't really see any major fatty acid metabolite imbalances, but these first two guys right here are indicating what was the case in this case was severe malabsorption. Methylmalonic. This is a marker in the nutritional marker section. Has an asterisk next to it, which basically means that when methylmalonic is high, it's indicating a B12 deficiency. Now, the metabolic pathway for B12 is complicated. The metabolic pathway for the production of methylmalonic is also complicated. It's interesting in that one of the things that helps to move things in a forward direction in this pathway is biotin. One of the markers that's seen on the organic acid test for a biotin deficiency is an elevation of something called methyl citric. It's actually on the nutritional markers section of the OAT. But we need adequate cobalamin as B12, okay, in order to help us make adenosylcobalamin, which eventually helps us make succinyl coenzyme A. So if we have a deficiency of cobalamin, or I should say a deficiency of B12, well, we can start to see an increase of methylmalonic acid. And that might just show up on somebody at a low level, they don't even know it, and somebody else that may be highly, strongly correlated to memory problems, to dementia, to fatigue, attention focusing issues, etc. But if we take this a little bit deeper and start to wind this information and see how we can link things back to other areas on the OAT, well, methylmalonic acid creates something called methylmalonyl coenzyme A, and in the presence of B12, the adenosylcobalamin helps to convert methylmalonyl coenzyme A to succinyl coenzyme A. So where do you think this is going? Okay, where is succinyl coenzyme A heading? What happens if somebody is B12 deficient? What's an area in the body that can become, or I should say an area in the cell that can become compromised. Right back into the mitochondria. Succinyl coenzyme A is another point within the Krebs cycle of our mitochondria. Now it's interesting, one of the cofactors that's needed in the metabolism of alpha-ketoglutarate or alpha-ketogluteric acid is thiamine. On the last page of the organic acid test, under the amino acid metabolites, there are some markers. They don't show up all that commonly, but they do show up from time to time, that are indicative potentially of a thiamine need, perhaps a deficiency. There are some genetic diseases linked to these particular pathways. Usually in the genetic disease condition, the levels are extremely high. Well, thiamine is an underappreciated nutrient in its link to many different types of chronic health problems. Dr. Lonsdale is a metabolic biochemist who's written a lot about thiamine deficiencies. And if you remember back to medical training, beriberi is this very severe form of thiamine deficiency. It can lead to cardiovascular collapse, respiratory problems, nerve damage, weakness, paralysis, all brought on by a deficiency of thiamine. Encephalopathy can occur because of thiamine deficiencies. Excessive alcoholics are at risk for thiamine deficiencies and some of the brain abnormalities that occur with thiamine. People can develop what are called paresthesias, these abnormal sensations, pins and needles, for example, in their extremities. But we know that thiamine deficiency could also be a contributing factor to mitochondrial dysfunction. It's been shown to impact the limbic system, which is very much involved in emotional control. Something I see a lot in the autism population is emotional imbalances that oftentimes, or I should say sometimes, are improved by thiamine supplementation. Autonomic nervous system can be impacted because of thiamine deficiency. So the markers on the out, even though they may not indicate a disease state, could certainly indicate some type of dysfunctional state where thiamine supplementation could be worthwhile for a wide variety of people who, again, are dealing with imbalances as opposed to a disease condition. Because functional medicine is, again, about looking and trying to optimize normal function of the body and trying to get an idea of, well, how's the body supposed to function and what can we do as functional and integrative medicine practitioners to try to help that person overcome that, either that imbalance or that deficiency. So as you can see, the organic acid test, again, is more than just one test. It's multiple tests, okay, placed into this very comprehensive profile. And I often look at the organic acid test like an onion. There's a surface level, and the more you peel off, the deeper you go. And it takes time to learn how to use the test and to learn how to prioritize the information. But you don't have to spend years and years and years, trying to study the test before you can start using the test in practice. But you do have to think critically and think clinically about how the test applies to your patient. It's not just about supplementing everybody for every single abnormality or level on the test. It's about being very specific with regards to what you use and learning how to line things up clinically. As I mentioned before, to me, the ode is kind of the hub of the wheel. It's a focal point. It's an entry point into functional medicine testing. It's, in my practice, nobody gets away with not doing the ode. Okay, it's a requirement. You got to at least do the organic acid test because we can initiate a lot of intervention just based off this information. And from the organic acid test, we can use other tests as complementary tools, the GPL tox profile, the mycotox profile, glyphosate, etc. As I mentioned in the beginning, we're going to be launching a new course called Advanced Oat Mastery Course coming up October 23rd. And by the way, there's a typo here. It's Advanced Oat Mastery Course. I didn't see that when I put the slide together, .com. And this course is going to be very comprehensive. We're going through every single marker on the organic acid test, as well as prioritizing categories and taking this level information to a whole new level of understanding and from a clinical standpoint. And we're going to be diving into the biochemistry too. If you came on late, you can text the word OAT, O-A-T, to 66866 and get access to this special report. It's a complimentary special report on understanding the OAT. So OAT, text the word OAT to 66866. I mentioned early on we had released a notice about this special report. Some people were having access. What ended up happening is the website that actually has this book, for some reason, disappeared. The web page did. So it got put back up. Anyway, that's just the nature of the internet sometimes. So OAT to 66866. If you're needing support in lab testing, there's a website called labtestplus.com. This particular website carries many tests from Great Plains Laboratory, including the organic acids test and other tests as well. So you can go to labtestplus.com and get a full profile of what labs are offered. I'm also available for private patient consultations through my private practice. MySunriseCenter.com is the website, and you can email me, email to scmedicalcenter@gmail.com. And then we're also launching a new service through our practice. Both my partner and I, Dr. Trincatella, who's my partner in practice, she's a naturopathic physician. Has been doing functional and integrative medicine now for, over 20 years. This health practitioner consultation services for licensed healthcare practitioners who are wanting one-on-one mentoring, assistance with lab interpretation, clinical correlation, troubleshooting different cases, supplement medication options, and other relevant information from an ongoing educational standpoint. So this is a new program that we're implementing through our practice. You can contact our practice at scmedicalcenter@gmail.com for more information. We are adding an information page to our website for this. It's not up just yet. So that's coming here soon, but you can contact our office directly for more information. That would either be consulting with me or Dr. Trincatella, and we're going over a wide variety of different tests. So all of the Great Plains lab testing and other tests as well. All right, everybody. Thanks so much for your attention with this webinar. Please reach out to us if you have any questions. If you posted any questions, Great Plains will generally, what they'll do is they'll email those to me within a day or so, and I can reply back via email. If you want any more information about our mentoring service, the courses, you've got the information there as well. Okay, so this is Dr. Kurt Woeller. Thanks so much. Take care.